Well, good afternoon, everyone. Sorry I'm a little bit late. This campus is getting bigger and bigger, and it takes a little longer to get across things than it used to. But again, good afternoon and welcome to everyone to the first Principals Research Award lecture. And I think everybody knows me here, but in case you're wondering who I am, my name is Franco Vaccarino, and I'm the principal here at UTSC. I'm privileged to be the ninth principal of this wonderful campus. Let me tell you a little bit about today's event. In the early 1990s, our campus leadership had the foresight to establish an award that, first of all, recognized outstanding research achievements, and secondly, supported leading-edge scholarship and research activities of UTSC faculty members. Now, since its inception, 22 faculty members, distinguished researchers, have, through the award, been recognized for their outstanding achievements. And I have to tell you, the awardees have covered the full spectrum of scholarship. I'm going to actually go through them. We've had zoologists, chemists, political scientists, management and business faculty members, mathematicians, anthropologists, historians, psychologists, artists, and biologists have been recognized by this award. And I think, actually, one of them is here today, Professor Friedlander, our mathematician awardee, and also the recipient of many other distinguished awards. Great to see you here. Our research enterprise here at UTSC continues to evolve and flourish, and I have to say we're attracting some absolutely outstanding world-class faculty whose scholarship and research really does have far-reaching influence and benefits. In many ways, the comprehensive research-intensive university campuses like ours really have two identities. We're an educational institution, but we're also a research institution. And this second identity of our campus continues to evolve as we continue to build UTSC as a destination of choice, not only for innovative and forward-looking programs of study, but also as a research destination of choice, as a place where research excellence is promoted and the spirit of scientific inquiry is applauded and encouraged. And I do have to say in this regard that I'd like to offer my special thanks and gratitude to Professor Malcolm Campbell, our Vice Principal of Research here at UTSC, for really being a wonderful research ambassador for the campus and for his leadership and really helping to take our research enterprise here at UTSC to new heights. So thank you, Malcolm. Now this year marks the first year that we're having a Principals Award lecture, and I have to say that all credit here goes to Malcolm in his role as Vice Principal of Research, who saw the benefits of having the annual awardees share their stories, share their journeys in the form of today's special lecture. And I should also say that I'm delighted to see that Professor Kronzucker, who will be formally introduced in a moment, is this year's award recipient and having him here as our inaugural speaker. And his really is a wonderful journey and a great story. So on that point, let me pass it over to Professor Malcolm Campbell, who will do the official introduction for today's inaugural speaker.
Thanks. I find it somewhat strange to be introduced by Franco as Professor Malcolm Campbell. Usually my mother is the only person who introduces me as Professor Malcolm Campbell, but that's a matter of pride. It gives me the greatest of pleasure today to introduce our speaker, the recipient of this year's Principal Research Award. And our speaker today is someone who epitomizes the notion of quality and quantity, or quantity and quality, depending on which order you like to think about it. And I know that Herbert likes to think about both of those things. So Herbert Kronzucker is the recipient this year. He is our speaker today. And Herbert, as I said, epitomizes the notion of both quantity and quality. And he's a real stickler for those things, actually. Now, before I delve into why he epitomizes those two things, I want to say that the person who nominated him, who is here today, Greg Vandenberg, who was the chair at the time, indicated that he could imagine a person no better to be recipient of this particular award. And there was unanimity around the table when we evaluated this year's award nominees that Herbert should be the recipient. He is absolutely a perfect person to be having this award. So, quality and quantity. Herbert got his PhD in 1995, emerging from the lab of Tony Glass at University of British Columbia. And in the almost 20 years, not quite 20 years, since his graduation from his PhD, Herbert has published no less than 80 papers. Now that's a remarkable accomplishment for any plant biologist. In fact, it's a remarkable accomplishment for any biologist, and indeed for a good number of scientists. But it's not just that quantity that is remarkable, it's the quality of that work. And that quality is measured in any number of ways, the way in which that work has had impact. The work has been cited almost 4,000 times, so just a little over 3,700 times have those 80 pieces of research been cited, which is really quite spectacular, as is Herbert's H-index. Now, most of you in this room probably have no idea what an H-index is. Herbert is one of those people who cares incredibly about what an H-index is, so I will share with you that he has an H-index of 34. I don't know what way to try and share the notion of what 34 means, but it effectively, you've probably heard the saying, standing on the shoulders of giants. Well, Herbert is the giant upon which other people stand. So 34 is an absolutely gigantic number at any stage of one's career, let alone someone who is less than 20 years out of their PhD. So quality and quantity. Herbert's work, which he will describe to us today, is something that really lends itself to people citing it, to people being interested in it. Probably some of you did an experiment when you were a biologist in a high school class where you took a stalk of celery and stuck it in a glass of water that had food coloring in it. Anybody do that experiment? Yes, so a good number of you have done that experiment. Imagine doing that experiment and watching the way in which the food color moves through that celery stalk and understanding the intimate details, the exquisite details that allows that food coloring to move through that stalk. To understand at a molecular level how ions are being transported from one part of the plant to another. An incredibly important question which Herbert will talk to us about in more detail today, or at least I'm assuming you're going to talk to us about in more detail today. A little bit of science for us. I should point out that in addition to doing this work that really resonates with people, he explains it, he expresses it in a manner that really illustrates the quality of his work in a manner that is absolutely adored by anyone who listens to him, and particularly our students here at UTSC. For not only is Herbert an absolutely amazing researcher, but like all of our professors here at UTSC, he is an amazing professor, someone who professes his interest in science, someone who shares their interest in their subject matter. And I'm going to finish by illustrating that point and really teeing Herbert up and setting a bar very high for him, I hope, in the talk that is going to ensue. I've already expressed to you a quality measure, and that is the H-index, and that is one that's established by peers around the world. But of course, there's no better measure of success for a professor than ratemyprofessor.com. and I would like to read to you one of the many, more than 20 pages of comments on Professor Kronzucker can one find on ratemyprofessor.com. But I particularly like this one. I feel honored to have had the opportunity to learn from Dr. Kronzucker. Halfway through the semester, he has inspired me more than I believed possible. He is undoubtedly the greatest intellect I have ever encountered. I hope nothing more than to be as knowledgeable as he is someday. The great block caps, the greatest professor at U of T, and a truly remarkable individual. It gives me the greatest of pleasure to present to you today a truly remarkable individual, Herbert Kronzucker.
You can use your slide, Malcolm.
I'm sorry.
You still want to use it?
We can see.
Can we skip it? All right. So audiovisual, how do I get my presentation? Where is he? There. Yes, I actually have no idea what I'm going to talk about, so this doesn't really matter. Do we have slides? Do we not? I pulled up a chair earlier. Maybe I was going to pull a Clint Eastwood, I thought. I might have to do this now. There we go. There we go. Let's bring us to the first slide. There, yeah, very good. Thank you. I'll spare you the Clint Eastwood. So, it's a great pleasure for me to be given the opportunity to talk a little bit about what we do. I will not try to bore you to tears with data. That's what scientists do. We gather data, we show bar graphs and circular graphs and correlation graphs. I'll show you a few, not too many. Mostly it'll be a story about my journey that started way back when I was a little boy in Germany to become a plant biologist with a few detours to various areas of interest that have equal instillment of passion for me. And I'll try to get across concept. What exactly are the big research questions we examine in our laboratory? We'll move through some forests and we'll move to the issues of world hunger. But before I do, there's a few prelude slides I wanted to introduce. And one of them is a summary, well, by a famous philosopher, but it is really just a paraphrased version of about the only useful thing I ever learned from a relative. Family members tell you many things, you know, brush your teeth, lower the lid on the toilet. It was my grandfather who used to say, my grandfather who used to emphasize very strongly, when you see a flock of people walking in one direction, try to walk in the other. Try to be different. Try to be individual. Try to have the courage to be different. And in that, you're going to find freedom. It was also my grandfather who was the only one who endorsed my journey away from my home in Germany to Canada. He said, I know you have to do it. Do it. And he said that the day before he died. So this is one of the summary slides with which I want to start. To find yourself, you have to think for yourself, not to think like others do, not to be a follower, but try to be different. And this slide is mostly for the benefit of our students more so than my colleagues. My colleagues know this very well. Here's another kind of summary like this. I don't like to be inside a box. I particularly like this slide because this is a sandbox that is actually on a sandy beach. Some are allowed to play on the sandy beach, others have to be inside the box. Life provides many boxes, in many shapes, in many sizes, and so does science, so does academia, provide many boxes. And I've often struggled, our team has often struggled, with others trying to tell us that we're supposed to play in here, not out there. Sometimes the university does it too. It has a certain expectation of you. It wants you to be in here, and then you start shouting whilst others are allowed to play freely. There's Malcolm over here. Huh? Yeah. So thinking outside the box, critical. And one of the experiences that I have had in academia is that very often we're not so different from the Vatican. We're not so different as academics from cardinals in equal attire flocking in the same direction. There's one black sheep here, but even he's walking in the same direction. So very often we're similar to those. And for me, my sympathy has always been more with the real cardinals, the ones that are out in the wild, that are wild and free. They might be a little bit lonely up in the tree, maybe a little cold every now and then, but they're wild and free. Those are the cardinals that we should remember. This is important at a university because very often we try to behave. Remember, Malcolm's presentation was about behavior. Behave. Right? So Herbert will not behave. And an institution like ours would benefit, as science indeed in general would benefit every now and then from a little lightning strike. This actually happened when Pope Benedict announced his resignation the day after the Vatican was hit by lightning. I really like that image. We need this every now and then to wake us up a little, to jolt us up a little, so that creative thought can fly the way the cardinal flies. To think vibrantly and to think fluidly is not possible in a box. And it is not helped by institutional constraints. So those are the introductory slides that I wanted to bring up. As a plant biologist, this is part 2, you have a lot of explaining to do. I always have a lot of explaining to do to my students. A few of them are here. Kevin will remember when I gave my presentations to hundreds of undergraduate students. And the typical question is, why did you end up studying plants? Why would anybody in their right mind end up studying plants? I know Greg gets the same question all the time. When we do surveys in our first year courses, second year courses, put your hands up if you want to go into plant biology. How many hands do you think will go up? No, of course, this many. None. None. In a lecture hall of 500 in the ARC, none. If you ask a question, how many of you have a dream of going into medicine one day? Almost every hand goes up. I have numbers on this, 85 to 92% year after year of our students answer in this way. I'm in a biology course because I want to be a medical doctor. Certainly not this. So you have explaining to do if you study plants, if that's the choice you have made. Now, for me, it doesn't take a lot of explaining. The tallest organisms on Earth are plants. This is one of the tallest trees ever measured in Humboldt County in California. There are eucalyptus trees in Australia that are even a little bit higher than this one. Animals can't do this. This is one of the most massive organisms on Earth, weighing about 2,000 tons in biomass, and it's a couple of thousand years old. You can drive a truck through it. How many animals can you drive a truck through? And they will still be alive, breathing, photosynthesizing. They'll still be here tomorrow. Name one. Can't do it. This is one of the oldest trees on Earth. It's about 5,000 years and counting. It was already ancient when Jesus was around, when Buddha was around. Now, if this does not inspire awe, then I don't know what will. Our students, however, don't seem to get it. Now, I'm from Germany, so it's easier to understand. We're the Teutons, the Teutons. The Teutons are named after the Teutoburg Forest. This is a famous battle in Germany, one of the few that the Germans actually won. About 2,000 years ago, this is a general, General Varus from the Roman Empire, and he was trying to invade the Germans in their dense forest empire, and he couldn't handle the forest. The general couldn't, and he lost bitterly, losing 3 legions, and it was bloody, and the Germans had been known ever since by the Roman Empire as the people of the forest. Trees. We don't want to venture in there. There could be Germans hiding behind them. So for me, having been born and raised in Germany, being fascinated by plants, and in particular by trees, didn't take a lot of imagination, really. It was a bit of a downer coming to North America, having to learn that most people aren't that interested in trees. But there are much better reasons to be interested in plants from a modern scientific perspective. One of them is that the biochemical sophistication of plants is absolutely stunning. Plants are capable of making hundreds of thousands of chemicals that animals can only dream of. Not only, of course, can they live on their own by photosynthesizing, pulling carbon dioxide out of the air, making all their food, requiring a little bit from the soil in terms of water and nutrients, that's it. And then they make all this magic. No, in addition to this primary metabolism miracle, plants perform what we call secondary metabolism and produce incredibly sophisticated chemistry. Chemistry that most people in laboratories, however sophisticated they might be, with degrees from Stanford and Harvard and Yale, cannot reproduce. I show an aspirin bottle here because the first aspirin ever used was from the bark of willow trees. And I still use this on hiking trips when I have a headache. I look for a willow tree and I munch on it because it's got a natural version of aspirin in it. More than 50% of all active ingredients in in medicine today are derived from plants. We would have no weapons against childhood leukemia or Hodgkin's lymphoma were it not for the biochemical sophistication of plants. At the cellular level, plants are far more sophisticated in their organization of the cell than any animal you can name. The genome of plants is far larger than that of animals. A chicken has about 16,773 genes, under 17,000. If you go to a rice plant, it's got 41 to 61,000 genes. Far more sophisticated genetically, biochemically, and at the cellular level. Humans only have about 24,000 genes. A rice plant has 41 to 61,000 genes. Remember that. Even a grape has more genes than a human. So don't you think, just because plants are sitting there looking a little bit stupid, they're not doing much, that they're not incredibly interesting from a scientific perspective. To a biochemist, to a geneticist, to a physiologist, they are a playground not to be rivaled by anything else. But of course, first and foremost, plants are the solar collectors that bring the energy from the sun into the entirety of the biosphere. None of us would be alive without the ability of plants to capture that solar energy. All this hoopla we're hearing about solar collectors and photovoltaic cells and the great successes there. Plants have been doing this for hundreds of millions of years. And how? With an efficiency, an incredible efficiency, that no engineer can rival to date. The solar collectors produce, of course, also all the world's primary food. In fact, all the world's food. Even if you say, no, I don't eat any grain. I eat chicken. I eat beef, and so on. Well, the chicken and the beef had to eat their plants first before they... All the energy, all the carbon that enters the biosphere and makes it onto our dinner plate comes from the primary photosynthetic activity, the miracle in the chloroplasts of plants. That's a fascinating realization once you really think about it. The other thing you have to realize is that there is a limited commodity of that photosynthetic activity on the planet. It's not limitless. We have a limited amount of land area on planet Earth. 71% is ocean. There's some algae here. They also perform photosynthesis. Almost all of that photosynthesis is along the continental shelves near the poles. As all my undergraduate students will remember. Kevin, how much photosynthesis? Numbers, please. Now, he's in a business program now. It's been a while. You'll notice areas that are darker green and others that are quite non-green, quite pale looking. Well, obviously, that's the Saharan desert. There are many areas of the world that are not good at supporting plant activity for photosynthesis, and others that are really, really good, like the Congo Basin, like the Amazon Basin in Brazil. So there's a lot of miracle going on, but it's limited. And we're going to come back around to this later on in terms of how much human population can be supported by this. Now, this is a graph I show in all my ecology classes, but I also show it in all my public lectures, because there's lots to be learned from it, lots to be remembered. Even if you don't believe any of the justifications and validations for being interested in plants that I just gave you, you will have to understand this foundation that plants truly are the foundation of all of life, that everything that crawls, that slithers, or flies got its energy from plants. And as those organisms do get the energy from plants, it gets whittled down and whittled down. When the herbivore, the one that eats only plants, eats those plants and walks around and talks and makes love, has a life, it respires away most of that energy that it took in from plants. As a rule of thumb, some 90% of that energy that was contained, that was so laboriously put together by plants, is lost in the journey to the herbivore. In other words, the cow or the mouse that eats the plant material will only retain about 10% of that biomass, and will have puffed some 90% of all the hard labor into thin air in the form of carbon dioxide and dissipated energy. And so it continues up the chain, the food chain, so that ultimately you run out of energy. That's the nature of the trophic pyramid. And it must be remembered and consolidated. I don't think we talk about it enough, that it starts here. And anything that is a constraint down here will limit our ability to feed the rest of the food chain, including humans. Now, plant biology in modern time isn't just running around identifying a plant, giving it a Latin name, not just observational. It's incredibly experimental. It's become incredibly sophisticated, in keeping with the sophistication of plants' biochemistry, of their cell biology, of their genetic complexity. It's become a true science with very, very difficult to understand and maneuver machinery. So here's a plant biologist working on this thing, maybe an animal biology, could be a chemist. They're all seemingly in a very similar realm of sophistication, and they most certainly are. For me, simply running around and looking at plants, identifying them, is fun, but would not be sufficient to turn me into a plant biologist. But when I was in my teenage years, there was enough of this plant biology going on to suck me right in. And it goes back to a picture I saw, I found it again, in one of the books of my father's, whose tie I'm wearing today, of Marie Curie, one of the greatest scientists who's ever lived. this very picture was in a book my father had. They had pictures of the Titanic sinking and things like that, big historical events and important historical figures. And this one really drew me in. I don't know why it did. I was about 8 years old and I kept looking at this thing. I like it. I like the way it's lit. I like the face. There's an intensity about it. There's an honesty about it. This isn't fake. One day when I grow up, I want something like this. Obviously began settling in my mind. And modern plant biology fulfills this kind of requirement of reaction flasks, of tubes, of sophisticated machineries, of laboratories that are necessary to unearth answers beautifully. Unfortunately, I was also interested in other things. And I remember maybe not so unfortunately, I remember very well at the age of 13 turning on the radio. My parents only listened to German Humtata music, that kind of thing. But I turned on the radio and I switched to the wrong station. And on it was a young Gidon Kramer from Russia playing the famous concerto by Pyotr Ilyich Tchaikovsky, the D major violin concerto. I had never heard anything like this. I played guitar at that time. I was very much into music. I parked the guitar in the corner. I worked construction for the summer so I could afford a violin. I wanted to play that music. This was the first time that I was really pulled out of all the other interests. This was it. I wanted to be a musician. So blame Gidon Kramer. Nevertheless, what does a confused soul do that is inspired by science, by music, and by so many other things? Well, I studied a whole lot of things. When I went to university, I enrolled in biology. And how would you symbolize biology like this? Well, because for us, it isn't just genes, flowers. There's a lot of mathematics involved. I'll show you later on in the kind of biology that we do. I enrolled in chemistry. I, of course, enrolled in music. And I also got a medical degree in passing. In Germany, that's possible. You don't have to do an undergrad first and finish it all and then become a medical. No, you can actually get into medical school right away. I couldn't decide. All these pulls were there, influences from parents, father saying you got to do this, certainly not that, that's not secure, right? So there was a lot of confusion, and it now required important influences, personalities to define direction. And those came in the form of these two somewhat encrusted looking fellows. Two of the greatest scientific minds I have ever met, bar none. One was a professor of ecophysiology at the University of Würzburg, Otto Lange, studying bizarre organisms like lichens that grow in bizarre environments like the Negev desert. How do they survive under those extreme conditions, he was asking. And he found beautiful answers to these questions. Otto Lange, what an intellect. What a stimulating series of lectures. I remember every word he said, I swear to you. And then a second one came along just as I thought it couldn't get any more exciting than this, and it was Ulrich Heber. Ulrich Heber, a photosynthesis biologist who was concerned exactly with this process I showed you the solar collectors for, the capture of solar energy and carbon dioxide out of the atmosphere to make all of biomass. And he was interested in the light reactions of this, how the electrons are jumping from one place to another, and how they do this on the nanosecond time scale. I thought this was cool. I thought it was the coolest thing. I had to enroll in this lab. I canceled medical classes, only attended those sporadically. And then I became one of his project students. Did a beautiful project with him. And I could never get out of this environment again. So it was two plant biologists who had so much enthusiasm, who were so fantastic as individuals, welcoming students into their labs, and just had an understanding of biology and life that I wanted to also develop as I grew older. They reminded me of these kinds of images. Now, advanced in their ages as they were, they had the curiosity of children. Anything that came around that was interesting. I remember the first day in Ulrich Heber's lab when he showed me his apparatus where he measured these electron reactions. Something went wrong and he opened up the machine in the back and he fixed it up a little. Oh, now look at this, he said. Look at this. When the first trace came out. The guy was 64 years old at the time. He's now over 80 years old. He just won a huge award in Germany for his scientific lifetime contributions. Both Lange and Heber are, of course, ISI most widely cited scholars. They have transformed our way we think about plants, ecophysiology of plants. But they remind me of this, even though they're encapsulated in older bodies with gray hair, thinning hair, they retained and cultivated and communicated that enthusiasm. And that's what I wanted. I felt it. I knew it. That's what I needed. I found it in two places only, in music and then in the plant sciences. Well, music, my biggest influence is right here. This was my violin teacher, Professor Nakura, founding member of the Tokyo String Quartet. So at the same time as I was in Ulrich Heber's lab working on electron reactions, I was still faithfully going to my violin lessons and trying to learn this damn Tchaikovsky concerto. It's not easy. But she did something else for me, and one thing really led to another, and that's why I'm mentioning music here. Not only because both science and music are rafts of passion for me, and sometimes one wins over the other. Most of the time music wins, actually. But it is both. Both are rafts that are floating very powerfully. It was Yoshiko who proposed me for a scholarship to go to North America and participate in a summer festival in Aspen, Colorado on violin and practice with a very famous violinist that I couldn't pass up. And I did this, and this was my first touching base in North America, and there was no turning back. And it was an invitation to play this piece by Niccolo Paganini, I Palpiti. I practiced like a fiend. This was a period of my life. That's the composer. By the way, in contrast to the Vatican earlier, they didn't even bury the guy in a graveyard. He was supposed to have had a pact with the devil, and therefore he was not to be buried on holy ground. This was rectified later on, but yeah, Niccolo Paganini, one of my great heroes. This is sort of a symbol of this, this time of my life when I came to North America. It was an ascending scale. It's been downhill ever since. Not quite, not quite. And I had the enormous opportunity and privilege to study with this man that I keep seeking out today, and that's Maestro Pinkas Zuckerman, who is now, in fact, the National Arts Center of Ottawa artistic director. A colossal intellect and a colossal violinist. And I had a chance to study with him in Aspen, Colorado. What a chance this was. So there was a danger at that time I would be pulled right back into music and full-time and not scientifically, but not quite. I went a little bit too fast. There we go. The mountains around Aspen, Colorado are rather beautiful. This isn't just a great place to play music, and it's actually great to hike through when the music festival is on in Aspen. You hear people playing trumpet in the corner, and there's a little violin playing Galamian scales. But I also went hiking, of course, hiking through the mountains, and was quite fascinated by this mosaic of colors in front of me. And I wanted to get an understanding, a mechanistic one, why these yellow trees were here and these dark patches of other trees were over there. Why? It's the same mountain range, the same valley, the same aspect. Why such a mosaic of colors? So I did strange things, and I ran around with rosin containers, rosins for the bow and the violin. I had nothing else to collect anything in. So I asked everybody for their rosin containers to put some soil samples into the rosin. So I ran around my hiking trips and I took soil samples from under those yellow ones. These are trembling aspen trees. And did similar things from the darker trees over there. Those are Colorado blue spruces. And took those back home to Germany with me to do some analysis later on, much later on. And when I did this, amazing things emerged. Here's a close-up of these trembling aspens, and then some spruces back there. The following emerged. Actually, many things emerged, but the most important, significant differences that came out of my rosin containers with the soil samples was, whoa, every time I saw these hardwood trees that are either yellow or green, by the way, this is one organism right here, just many stems, one clone. Malcolm knows a lot about these kinds of poplar clones. Trembling aspen, that green one is another one. In fact, it's by their color demarcation that you can tell it's one organism. Some of these aspen clones, by the way, if you weren't fascinated enough earlier, are some of the oldest and largest living organisms on Earth, clonally speaking. This particular one is about 60,000 years old, but there are others that are much older than that, 80,000, 85,000 years old, for the same clonal organism. Yeah. Anyway, so there they were, and as a chemist as well as biologist, I was interested in what was in that soil. And this difference emerged that nitrogen appeared in two forms. One was in the form of NH4 plus, or ammonium, the reduced form of nitrogen, and one was the oxidized form of nitrogen, nitrate, and along with it, there were different bacteria. These are bacteria in a close-up in those soil samples. I brought them all the way. I was probably all against regulation to bring non-sterilized soil samples, but this was back in the 80s, with bacteria in them, with nitrogen and ammonium in them. But those differences, they shook out every single time. Every rosin container full of soil from under trembling aspens had NO3- and corresponding bacteria in them that produced the NO3-. And every single rosin container with soil from under the spruce trees that are really dark here, there's a bit overexposed this picture, had ammonium NH4 plus nitrogen in. Now this isn't trivial because nitrogen is the most important food, the steak, if you will, that plants require from the soil. Nitrogen is the most often limiting factor to the growth of plants. So other than the carbon that comes out of the atmosphere and the water that comes from the soil, nitrogen is the third thing in line that plants require to thrive, to grow fast, to produce the biomass that has its constraints built upon it that we talked about earlier. So this fascinated me and I wanted to learn more about it. Why nitrate here? Why ammonium over there? Is there something about this soil chemistry and biology and the resultant nitrogen cocktail that allows the growth of some species over here and disallows their growth over there and vice versa? So I became very interested in this question and in so doing stumbled upon a realization of the Buddha. The simple realization that, and we often don't get this, nothing is stable. Everything is constantly changing. Everything is in flux. In fact, this is so misunderstood that every evening you can switch on the news just about and hear something else about climate change. Ooh, the climate is changing. The world is changing. Or articles in newspapers that say, in a changing world, ... Yeah? Is there any other kind of world? There is no other kind of world. It always changes. So this realization was very important from this perspective of the picture I just showed you, that ecosystems as well, as we stare at them, take snapshots of them and count the plants that are there, give them names, look at the chemistry in the soil. Those are just snapshots in time. What if I came back to that same site 10 years later, 20 years later? What would happen? Well, the history of that site that I was just showing you, with those nice yellow and light green aspens on the one side and the dark spruces on the other, had been harvested. It had been subjected to disturbance. Clear-cut harvest. The trees were taken out and it looked like this. And wherever it looked like that, the spruces that were there before and were taken away by harvesters, they couldn't come back. They couldn't grow anymore. This environment was too different for them, and those aspens that we saw, these clonal, these large, these colorful ones, they loved it. They came in, they invaded. And it's in these kinds of disturbed sites that the nitrogen chemistry that we just said could come in those two forms, nitrogen and ammonium, there are other forms, but those are the two most important ones, would be converted into this NO3 minus form that the aspen loves. In other words, the food that is provided now after a disturbance to the ecosystem disallows the thriving of the original species that was there and allows others to come in. To me, this was a big realization, and I wanted to learn more about it. In ecology, we call this kind of development succession. Picture I always show my classes when we discuss succession, how a recently disturbed, opened up ecosystem, so there used to be a forest and then they open it up to develop a field, develops over time when you leave it to its own devices. No more human interaction after the original disturbance. 7 years in, 15 years in, 30, 95, 150. It takes a long time, but eventually the forest does come back. At least in many cases it does. And this chrono sequence, that sequence of development, is called succession. And I learned that in the process of this succession, that very thing I was interested in was pivotal. That in fact nitrogen, this most often limiting growth factor for plants, transitioned from nitrate after disturbance to gradually more and more ammonium. So that in the end, when the forest was back in its complete ancient state, if you will, it was back to an ammonium soil and would be reconvertible to a nitrate soil were you to disturb the ecosystem again. Chronosequence succession. And we see this everywhere. Every time you go up to Algonquin Park, you see it. Anytime you see those vibrant fall colors of the maples in upstate New York, for instance, we see stages of that chronosequence. In fact, these are middle stages. This particular drive along Algonquin Park was completely harvested in 1830 to 1840, in that period of time. And since that harvesting, taking out the ancient conifers that were there, the white pines and the red pines, it's those beautiful maple trees that came back and took over. They couldn't help themselves. Well, the conifers couldn't help themselves. They couldn't grow there anymore. So here too, in our famous and beloved Algonquin Park, we see the signs of chronosequence, of change, that in fact, if we left it to its own devices, 50 years from now, a trip to Algonquin wouldn't be so pretty no more. Those fall colors would no longer be there, not in that form. The maples are only here as temporary guests. They will disappear. In some environments, like these, in California, we call this the chaparral environment, the return to the original ecosystem after disturbance is no longer possible. And we're locked into a quasi-steady disturbed scenario. What we now take for granted as the natural landscape of California is not natural. Where this cyclist is having fun in the Californian chaparral along the water is where there used to be some of the world's tallest trees, like the ones I showed you earlier. The Sequoia sempervirens trees and the Sitka spruces used to live there. But they were deforested, they were removed in such great abundance and so efficiently that the climate, the soil environment, everything changed so profoundly that there was no path back to where the original landscape was. Usually that kind of problem is associated with a large amount of erosion of the soil so that you cannot return to the original. This is, by the way, also the reason why in a place like San Francisco you have so much fog. This is water from the ocean that originally, prior to San Francisco and its environs being converted into this chaparral, shrub, grass environment, when it was still covered by trees, tall trees, that water had places to go. It was in the hydrological cycle of the forests. Now it's got no place to go, and you see it as fog. So again, the snapshot in time, change, should we be alarmed? Yes, change is happening all the time and visible just about everywhere. A little bit more from Algonquin, from a recent trip there. There too are some environments where recolonization by plants has not yet been successful. This is still naked rock. This particular piece, by the way, is 2.2 billion years old. It's quite intimidating when you sit on it, that's 2.2 billion year old rock. Plants have not recolonized it after the glaciers had their go at it in the last ice age. So this is now for some 20,000 years remained bare. Prior to that, it had kilometers of ice on it. But in most other places in Algonquin, you have plants that came in after the ice retreated and moved back in to reclaim the land and did it gradually. And once again, the nitrogen chemistry followed it in the ways that we saw earlier, except over a much longer time period. A few more images of plants struggling to become established. That's how they do it. Some hardy ones at the edge, they collect a little bit of soil. Here's one really struggling. A few people that are a bit too close to the edge. In fact, that day I was there, I thought, oh my God, this is going to fall down. But here they are, hanging on for dear life and just accumulating a bit of soil here. Gradually, a little bit more will blow in. This will take many more decades for there to be a proper coverage by plants. But in the process of that change, of that development, the chemistry in the soil changes with it. Same with fire. When fire rages through an ecosystem, it creates a new blank slate, if you will, of chemistry and biology that is now possible. And very often, the species that thrived before the fire raged through have no chance after. One of the great changers of ecosystem dynamics, fire. Here's a species that does well. This forest burned down. And this is a species that's adapted to fire, and up they come. This is lodgepole pine, doing very, very well. But a lot of other species cannot do this. What nitrogen source do you expect down there? It's very fresh. It's right after disturbance. Yeah, you guessed it. There's a lot of nitrate nitrogen there and very little ammonium there. And if you now study that lodgepole pine for its adaptation to that nitrogen source, it'll show you how much it loves it. I'll show you a few data about this. Now, I wasn't so much interested in lodgepole pine. Of course, here's the violin again. I was interested in spruce. Why? Because spruces make violins really what they are. There's a little maple underneath, but that doesn't really matter. You could put beech there. It doesn't really matter. It's the spruce that makes the difference. The spruce is the sounding board up here. The top of the violin is spruce. And most importantly, there's a little post that connects the top and the bottom of the violin. It's called the sound post. And in Hebrew, it's called neshama, the soul of the violin. And that soul of the violin, the sound post, is made out of spruce. So naturally, I was more interested in spruce than in pine or any other species, and that's what became my subject for studying root dynamics in this laboratory that Malcolm mentioned earlier to examine exactly this question. would there be adaptations to these two sources of nitrogen in spruces? Here they are, the ones that give you the sound post in the violin. There they are. So these ones are not that interesting. So that became my focus. Are they really adapted to that source of nitrogen almost exclusively, to the point that they can't use this one? So how do you examine such a question? Well, when I started in biology, there was a trend developing that has not abated since. It's a somewhat unfortunate trend. It has good sides, but it's also somewhat unfortunate. And that is to try to grind up plants into little pieces and then analyze what's in them biochemically or genetically. What kinds of genes are there? How are they expressed? Blah, blah, blah. Well, for me, this was not, it was obvious that genes are responsible. Genes are the script. But this is it's a musical sheet, but it itself doesn't make music. This is not music yet. It's just the script. I needed more than just looking at the script. Or, like I also like to symbolize it here with the Confederation Bridge, there's a large journey from the genome to function. And it's not an easy one, especially when it gets windy. This is where I wanted to be. I wanted to study the function and find ways in which it could be studied at a state-of-the-art level like nobody else did it. While so many of my biology peers were all here, they all wanted genome. This has some interest, and it does to me now, but not as much as the other side of the bridge. You must connect the dots. For instance, this is for the genetic crowd among you. How could you predict that this ballet dancer, a friend of mine, by the way, could jump like this from the genome? Couldn't. It's not in the genome. Remember Dorothy DeLay, the famous violin teacher at Juilliard, saying, "Don't think talent is inborn." It's not true. As long as you have an able body and a little bit of, in her case, musicality, you can be a great violinist. You got to work at it. Practice the muscles, muscle memory. Away, away, away. Never stop. That's how you become an expert. Not because your genes somehow predispose you to become a concert violinist or a principal at UTSC. It's not that simple. It's a question of decision making, of influences. You can't read it from the genome, but you can read it from a functional analysis when you actually study the living, breathing organism as it does its thing. Same when somebody has a headache. Do you think a genomic analysis would tell you? If you ground the person up and you looked at what genes are expressed and which ones are not, it would give you precious little information on the headache. Nor could you really deal with it therapeutically. Are you going to go in there and silence a gene when someone has a headache, or are you going to give them an aspirin pill? It's a very important principle that is not understood enough anymore. And in an extreme case, it's a joke I like to play on some of my colleagues at more esteemed institutions, there are examples where dead people cannot be identified as being dead by genetic analysis. When a person dies, there are cells in your body that are alive for several days. They haven't yet caught on what really goes on. And you could take samples of their genome and run them through PCR and figure out how they're you couldn't tell that they're already dead. So in other words, the genetic analysis is great, but it's often actually quite crude. It's not good enough to capture the refinements of the jumping ballet dancer or the disease or even demise. So I wanted to study function, following also the paradigm of one of my great heroes, Heraclitus, the obscure. There he is. He looks away. That's why he's so obscure. Heraclitus. Panta rei is what he said. Everything is in flux was the realization by this particular philosopher. This is very similar to the Buddha's realization. Nothing is stable. Stability is an illusion. Everything is in flux. So therefore, I thought to myself, the best way of approaching those research questions I had about spruces and nitrogen sources was to measure the fluxes of these nitrogen sources into and out of the plant. And I joined one of the labs that's the finest in the world for this kind of thing. And that's the lab of Tony Glass. He is receiving a big award a couple of years ago for his lifetime contributions. And here he is, a more recent picture that Dev sent me on as he's cycling through Cuba playing jazz. This is my former supervisor after retirement. Still publishing papers every now and then. And I learned from him the tricks of measurement of fluxes of ions into cells and between tissues And we did it in little babies of spruce. So here they are, the conifers, and we labeled them with radioactive versions of those two nitrogen sources of nitrate and ammonium, and try to figure out what the behavior of these nitrogen sources in the plant might be, how they flow across membranes from one source to another. This requires setups with lots of protection. There are a lot of lead pieces here to protect the workers from being irradiated to death. To my workers, this is a young Dr. Brito. There's Dr. Brito right there. Yeah, there he is. And there's Dr. Brito again, showing you that it's not just a question of what happens at the bench with this kind of work when you try to conduct flux analysis and get answers there about how things flow from one place to another. You also need mathematics and you need calculus, something I always have to hammer home to my students. Lasse, mathematics, calculus. Here's a little something that Dr. Brito and I came up with a few years ago. It's sort of our version of putting up a little gene chart, you know, A, T, C, Gs, and so on. Yeah, so there it is. That's as important as anything else. Here's Ming to embarrass him a little. We use approaches also that measure the flux of ions, not with radioisotopes, but electrically. And here's Ming, one of our stellar students in our lab here at UTSC, using an electrophysiology device with tiny, tiny electrodes that are inserted into the living cell to measure the electricity changes that happen there when an ion like the NO3 minus ion comes around or the NH4 plus ion comes around. Every time this happens, there's a little change in that electricity. And you might not fully realize this, but this is true for all living things. Every cell is like a battery. It's electrically charged. A little bit of a charge. It's about 1,000 times less than the charge through these outlets here, but they're all little batteries. And by going in there in a sophisticated way, we can use that electricity to measure exactly what the flux reality is for ions that we're interested in. Well, and this is what one of the big findings that came out of it for these spruces, published in Nature at the time, causing a lot of furore. This made it in the Encyclopedia of Life Sciences, and there was a lot of political storm around this because of the practical applications of it. But the science of it was really ultimately quite simple, and it showed us how for this ammonium source of nitrogen that was dominating in the mature forests that spruce loves, indeed, the uptake mechanisms in their roots had adapted perfectly to that food source, whereas nitrate was much, much, much discriminated against. This caused a lot of interest. And we used more refined methods of using radio tracing. Don't look at any of the details here, just to give you a bit of a visceral understanding of how this kind of physiological analysis allows you to literally take the pulse of the living, breathing organism. Not just grind it up and see what a machine gives you, but to take the pulse. This is how it's behaving in real time. So when we did this, it allowed us to, in fact, access what goes on inside the living cell. Is it possible that the metabolism of the cell, its storage capabilities for the two sources of nitrogen might be fundamentally different? And so they are. A huge preference for the ammonium source of nitrogen was seen over nitrate. And this doesn't look like anything special, but it was something special. This is something that had never been seen before. Almost all species looked at by researchers had always been exactly the other way around. They love the nitrate source. That's what a wheat plant loves, or a barley plant, even a corn plant. This is what a beech plant, a maple tree, that's what they love. But the spruces were completely different. And this offered an explanation how, at the cellular level, it was impossible for some species to colonize a site after disturbance. And for that reason, it caught on so much with the ecological literature that was interested in these succession processes after disturbance. Well, sometimes even plants can in fact take up too much. They can over-enjoy a resource. And this was the other side of the same story that we examined, looking at species that were happening early in the successional chronosequence as opposed to late. And again, the details of the graph do not matter. And even exactly what this means, except to say that it's the subdued version of this behavior that we measure in real time. That's the reasonable behavior. If you're up here, this is a logarithmic graph, you're an order of magnitude more crazy. This is the eating behavior, a reflection of the eating behavior. If you're up there where these early successional ones are, you are taking in far too much nitrogen, and because you are taking in too much, you're actually poisoning yourself from too much food. So plants do this too, so we looked at it from the other side of the story as well, not just the conifers that are so well adapted to ammonium nitrogen, that they like it, they take it in at reasonable rates and much higher than the rates of nitrate. But the other species that happen early in succession, like trembling aspen, actually were poisoned by too much nitrogen. Well, this work on nitrogen caught the attention of a completely different crowd that had nothing to do with forests. And that was the crowd at the International Rice Research Institute in the Philippines. They saw our paper in Nature and another one in a fairly prestigious journal, in particular on the behavior of NH4+. And so isn't that something? Your conifers that you like so much, they actually seem to behave like rice plants do. Rice plants are just as unique in the sense that they love this particular source of nitrogen. Spruce and rice, so distant from one another, seem to share a very bizarre behavior. And of course, this species isn't just any species, but it happens to be the #1 food crop for the human race. More than 3 billion people depend upon rice for survival. So I received an invitation letter to come to the Philippines and take on a Rockefeller Fellowship. And I remember this very well, arriving, jet lagged like crazy, knock on my door. Sun rises in the tropics at 6 o'clock, no mercy, and it sinks at 6. So people go to bed early, but they also get up early. I don't like getting up early. So 6 o'clock in the morning, sun rises, I get a knock on my door, and there was this man here. This is Gurdjieff Kush, the World Food Prize winner, the fellow who had headed up the rice breeding section of the International Rice Research Institute for 30 years, and was responsible nearly single-handedly for tripling the world's rice yield through his elegant approaches to the breeding of rice cultivars. There's some 120,000 such cultivars now. Right? So he knocked on the door and said, "Let's go out. The sun is rising." Okay. He had rubber boots for me. They were way too big, but so I put them on, and we went out into the rice paddies. You need rubber boots there, you really do. In fact, mine, large as they were not large enough. Before I knew it, I had mud all over my toes, and this isn't good, I should have stayed home. But I remember very well, about an hour into this, when the sun was really rising, and it was shining onto these oceans of green, of rice paddies in the Philippines, that my goodness. It was the first time in my life that I fully realized, even though I was basically raised on a farm, it's the first time I fully realized where the world's food comes from. It comes from these rice paddies in Asia. The majority of it does. The majority of the human race depends on this, and I wanted to work on it. And what a privilege it was, of course, to meet Gurdjieff, an incredible scientist, who convinced me of the urgency of this, that it wasn't just an issue of understanding nitrogen dynamics and uptake mechanisms. Here's this transporter and that transporter, and that's the rate of transport, blah, blah, blah. Yeah, that interests a scientist, but there was a much bigger mission. And that mission was to furnish enough of this #1 food source for the world's population. And just what a crisis this was, and is, I had actually not known. And most people still don't know. Most people don't understand how much scientific research was necessary to make it possible to feed the world's population today. And just because most of us don't go hungry, we go home and have a nice meal, that same truth is not the case in many parts of the planet. And I had to learn it firsthand, driving past the shacks in Manila and seeing poverty, real poverty, right in front of my eyes, and being reminded, oh yeah, the human population. This is the number as of this morning, basically. Last year, right around a year ago, we exceeded 7 billion people on the planet. I remember when it was half that much. That's me at that time when we only had 3.5 billion people. It wasn't so long ago that I was like this. And since that time, we've added another 3.5 billion people to the planet. And all of them need resources, and they need not just resources in the form of gasoline and space and houses, but they need food. And associated with that is the ability to furnish enough food for everybody, which is what we refer to as the carrying capacity in terms of food calories in ecology. How large is the carrying capacity of Mother Earth for the human population? Well, I'll give you just a few details about this. And #1 is we couldn't do what we do, which is to have 7 billion people nourished to the same level that we take for granted right here, were everybody on the planet to eat in the same way that we take for granted. It is not possible to consume as much meat as we do. You might not realize this, but look at the dark shades of red. Those are the heavy centers of meat consumption, also Australia, of course. The average North American, the average US American consumes 220 pounds of meat per year. In Canada, we're a little bit better, but also not very good. Remember the food pyramid from earlier, how the energy that comes from plants gets whittled down, how the herbivore will only retain about 10% of all that energy that the plants had. That's why we have the food pyramid. So the more we consume the meat and not the original plants that grew in the world's agricultural fields, the tighter the noose becomes, the more difficult it becomes to furnish enough calories to keep everybody well fed. So this crisis became very obvious to me. And you can calculate what the carrying capacity for Earth would be for human population were everybody on the planet to eat in the way that we do, with that kind of meat consumption, the milk consumption, the eggs, all that come with it. That's it. 2.5 billion people, no more. We would require nearly 3 planets Earth. That's the current evaluation, to support everybody in the way we take for granted. 3 planet Earth. This can't be done. It's not sustainable. Of course it's not sustainable. So I threw myself into this and have ever since, and have great, great interest in doing much, much more on this front. Of course, our starting point was nitrogen. This is where we came from. Here's a simple farmer throwing exactly that onto the rice field. Nitrogen. Why nitrogen? Why not something else? Well, because it's the most limiting nutrient in the soil. You can do more miracles with nitrogen from the soil than with just about anything else in terms of boosting production of plant growth in areas where you might have a certain yield to start with. You add nitrogen, you get more. You get more. And the miracles you can achieve are really quite astonishing in this regard. I mean, one of my technicians right over there, Wayne Wynn, currently growing plants all the way to yield, right here in Scarborough, rice all the way to yield. We're harvesting the rice. Is able to increase the yield of rice with simple tricks of nutritional physiology, many-fold. Many-fold. There's an experiment currently, more than 6-fold larger yield is obtained by understanding the biochemistry and the cell biology and tricking it into giving us more. So my early point in this regard was to mix up nitrogen sources, the two that I studied in isolation before. I brought them together, nitrogen and ammonium. Not changing the overall dose. In other words, giving plants exactly the same amount of overall food, just changing the chemistry up a little bit. And when that was done, a synergistic response was seen. The details of the graph of the table do not matter. I circle in red. These numbers here show how much more of nitrogen is taken in. These are fluxes. How much more nitrogen is taken into the rice plant compared to any of the other conditions. So even the most optimistic conditions, without doing this trickery, are significantly below the nitrogen intake rates that can be achieved, that are some 20 to 30% larger when changing up the nitrogen chemistry a little bit. The Rice Institute was very interested in this and is very interested in this, and there's a way in which you can achieve this kind of chemistry in the field by favoring certain bacteria in the soil that produce enough nitrate along with the ammonium. Here's a plant, exact same genome, exact same cultivar as the adjacent ones, but it's contained in a bag down there that has the mixed nitrogen source, as opposed to the other ones that have the single nitrogen sources. The growth and yield advantages are tremendous, and this is now used in many parts throughout Asia, in fact. And one of the ways in which you can achieve it is to dry up a rice paddy temporarily. Do not flood it constantly, but remove the water, let it dry up, and nitrate gets produced. And then you bring the water back, and ammonium gets produced again. So you play the chemical trickery of providing both nitrogen sources over time by staggering through an agricultural technique that came out of these physiological observations. Nitrate-ammonium synergism. But the other part that we were interested in is the loss of biomass. I'll show you the loss here for forests and agricultural systems, forests, and there's a cornfield there, from the original photosynthesis accomplishment by the plant due to its respiration. When the plant respires, does its thing, the plant respires too, even though it doesn't walk and it doesn't talk, it respires away most of the energy that it photosynthesized in the first place. And we were interested in to what extent that subtraction from the original potential of fixing carbon, of fixing biomass, of making food, was really necessary, and how much of it could perhaps be reduced down, could be dampened down, could we produce a more reasonable plant. And this came back in our example of ion transport people to the cost of transport. As everybody knows who's in economics, the cost of transport, the cost of transport. Don't just think about the final product, but think about the cost of transport. The cost of transport at the cellular level relates in plants predominantly to one enzyme. One enzyme, the proton ATPase and plasma membranes, consumes, as I like to say to my students, for every donut you consume as a human, about half of that donut goes to your ATPases. You have ATPases too. What do these ATPases do? Well, they're the ones that charge up all the cells to be electrically charged, the stuff that Ming measures with his electrical apparatus. That costs energy to charge these up constantly. And when there's a deviation, to recharge and recharge and recharge. And to move everything about, all the goods, glucose, sucrose, nitrogen, potassium, phosphorus, all the things that need to move, even those secondary metabolites like aspirin derivatives. Those have to be moved about. This costs a lot of energy. So as a rule of thumb, half of every donut you eat goes directly to this one ATPase in your bodies, and in plants it's no different. About 50% of its respiration goes to the proton ATPase in the plasma membrane. So we were interested in dampening that consumption. Would there be ways in which we could increase biomass? Again, the details don't matter. It's rice. There are several bars here. The black ones are important. The black ones show total biomass in the rice plant. Normal conditions, still conditions that had been achieved in agriculture, not done by anyone, and this is what we did. And we found plants that were now more than twice as large in their overall biomass than any of the normal conditions by using a simple trick that we had stumbled upon originally, and that was to optimize the supply of potassium, K+, another one of the macronutrients for plants. There's some 16 elements that plants require, and this is the second one in line after nitrogen, by mixing it at the proper ratio with nitrogen. And out of it came a super plant. We were very excited. The journal was excited, and we made the cover of this particular journal. But it's gotten a lot more exciting since, because we were asking, these are young plants. They're 3 weeks old, 21 days old, little rice babies. So what would happen if we grew them all the way to yield? Well, we've done this since, and we were in the 3rd round of doing it already. The gains are even larger when you look at yield. Very, very substantial. Many-fold more biomass can be achieved. How? By using the trick of potassium being able to suppress some of these fluxes. There are some of these unnecessary cycling activities that go on at the cellular level that we can suppress nutritionally and make it less of a donut consumption, if you will. More of the original donut remains. One of the ways in which this is done, just one example, is by stimulating one key enzyme. This is an enzyme that Greg knows a heck of a lot about, PEP carboxylase, that the interface of carbon and nitrogen metabolism, where carbon and nitrogen come together. We studied other such interface enzymes, and they're maximized along with this trickery of putting potassium in its right place into that ratio to nitrogen that makes it possible to produce a super plant. But more excitingly even than that, we stumbled upon ways in which we can reduce the demand of the plant for water. We all need to drink our 4 to 8 glasses of water a day, as we've all heard. By the way, that's a medical myth. Nobody in medical school ever tells you that. 4 to 6 glasses, what does that even mean? But yeah, you need a lot of fluids. Coffee's good enough. That's just a little life advice. Milk is good enough. Orange juice, it's not true that coffee dehydrates, and it's not true. You have to go to the bathroom more often, but it gives you enough water for your metabolism. So 6 to 8 glasses of water, that's not true. But plants consume water like you wouldn't believe. In particular, rice. For 1 bowl of rice that you might now cook up and make something out of, fry up and make something out of it, to get to that stage of being in that 1 bowl, about 500 grams of it, you require approximately 500 liters of water to get there. That rice plant in the paddy requires all that water. Most people have no idea that almost 90% of human water consumption occurs in agriculture. It doesn't occur in the toilets. We shouldn't flush as much, of course. Don't flush too much. But that's not where the principal consumption of water occurs. Not in the steel industry. There's a lot of water consumption there too, but compared to agriculture, it all pales, and in particular so in developing countries, in China, in India. India is closer to 94% in terms of its overall water consumption going to agriculture. You need to irrigate. And many crops are intensely thirsty, and unfortunately, the thirstiest of all crops is also the most important, and that's tropical lowland rice. That's why you see rice grown in fields like this, irrigated fields, just sucking the water in, sucking it in and sucking it in. And with that same trick that I mentioned with this optimization of potassium in relation to nitrogen, we can make the plant much less thirsty, go back to the previous graph, by targeting the molecular mechanisms in the membranes of root cells that are actually responsible for water transport. There was justifiably a Nobel Prize given out for the identification of these transporters in 2003 to Peter Augrey, the aquaporins, a family of proteins, of transporters that actually allow the cell to drink. This is how cells drink. All cells in all biological kingdoms, including humans. We need aquaporins to drink. There are tons of aquaporins in your kidneys, for instance, to make your kidney function possible, in your skin, etc., etc. And we found ways in which we can control the excess water flow through these aquaporins. That's something that we're working on currently. Devrim works on this hard with a radioisotope approach. He tries to bring it to bear on this. And Wayne has yield data for this that are quite fascinating in a new way. Not just is there a larger plant that has more carbon, more grain, but how much water did it require to make that grain? That water use efficiency we are able to greatly optimize by targeting these aquaporin proteins with nutritional players like potassium and nitrogen. And the other area we're interested in is the converse of it, that there's not enough water in so many parts of the planet, and very often these drought-stricken areas are affiliated with an accumulation of salt. Salinity affects over 80 million hectares of arable land on the planet, and we've been very interested in how plants are adapting to, dealing with these kinds of stresses in the field, with some spectacular results, the details of which I'm not going to reveal because most of them are not published yet. Last is sitting right there. There's a picture from the Globe and Mail cover article on this a few years ago. Some aspects of it, the Globe and Mail loved it, had a big article about it. Lots of interviews followed up. To cut a very long story short, the paradigms of salt stress, of how sodium is toxic to plants, has to be completely revised. So our team is in the process of doing this. Essentially what we're saying is it's not like people used to think that sodium is so toxic because it flows into plant cells at too high a rate. It overwhelms the cytoplasms of the cell. It poisons enzymes in those cells. That's not actually how it happens. It turns out that the flow rates for sodium into the living cell are tiny, tiny, just a little trickle. That's not how the toxicity comes about. There are processes going on outside the living cells in spaces in the cell walls of plant cells. This is one of the aspects of the complexity of plant cells compared to animal cells, that they have scaffoldings on the outside. They have cell walls. There's an entire research cluster about this at UBC that is world class, second to none, that studies just that. How do cell walls in plant cells behave? We don't even have them as animals. But it turns out that's where the salt goes. And that's where the salt flows. And that's where it builds up crystals. And when it does, it sucks water from living cells. It sucks potassium from living cells. And that's the kind of mechanism that we're trying to dissect very finely, in particular in Lasse Schulz's work currently. So there's a great interest in that salt tolerance that we have as well. And then looking into the future, yes, I'm almost done, going a little bit over time, we are trying to think up. Up, not just at the same level. You should always think up, always think creatively, as I said, fluidly and vibrantly, but also consider new options for agriculture. I'm a big supporter of what Dixon Pommier at Columbia is proposing to include vertical agriculture increasingly, in particular for high-value crops. But also I'm interested in, for food crops, such as rice, I like to sort of jokingly call it, when Malcolm and I talk about it, as high rice. You know, what is the potential of approaches like this that are not as land intensive and that could provide food in big population centers where most people nowadays live. They live in big cities, and yet we grow food far away under the vagaries of nature, salinity, too much water, too little water, not enough nitrogen. How can we adjust the potassium? In a high-rise construct, we can do all of this. We can create ideal conditions and furnish food. The only problem is to get the energy that's required to do it. But those are also things that interest us to come up with models, with calculations of feasibility of what it would require to grow food vertically in towers. So even things like this are entering our brains, and this all feeds into this discussion that Malcolm alluded to, our effort to have at UTSC a Canadian Centre for World Hunger Research. And we've worked on this for many years, great help of Greg Vandelberg over there, who's our chair, for the past 5 years, and Malcolm's support, Franco's support, and we've got a plan in place. That's so as we're thinking into the future, thinking up, if you will, that is what we're trying to do here. And I think we can make important contributions to this right here at UTSC to feed the world's billions. Thank you.
Absolutely wonderful talk. I'm going to open the floor for questions. Are there any questions from the audience?
Kashaf?
You said that rice plant loves chemical form of nitrogen. I think it is for lowland rice. How about the upland rice?
Good question. Is the preference for ammonium nitrogen mostly a lowland rice cultivar issue? Yes, it is. There are different cultivars of rice that you say are upland. They grow up in the high mountains. Many of them love nitrate or love mixtures of nitrogen sources. However, to global food production, upland rice is basically not a player. So it's of interest to a breeder, it's of interest to a physiologist, geneticist, but not so much to an agriculturalist. So we're not looking at upland rice in our laboratory. Everything we grow here at Scarborough is tropical lowland rice. The highest yielding varieties, the most important ones, the most salt tolerant ones, the most salt sensitive ones, etc. Those are the ones we've got here, right over there, science building.
Any other questions?
You talked a lot about soil and about water, but the high-rise structure raises the question of sunlight. I wonder if you could say something about sunlight in particular, about how that might be handled when you have a high-rise structure where obviously sunlight's not going to reach every level.
Yeah, that's the biggest challenge in any of these vertical agricultural approaches. Really, that there are two major ones. One is materials to build the building, and the second is to bring enough energy to the structures. Dixon Pommier at Columbia has published in Science magazine, various places, feasibility analyses for high cash crops, things that produce big fruit, like tomatoes or things like that. You can easily envisage avocados and those kind of things that fetch a high price per unit area to be feasible in such an environment to give you that return. You need to have artificial illumination, and you need to power it. Right? He's come up with models that say you could do it sustainably with wind and solar. I'm more skeptical on that front, but I would argue that the production of food is so fundamental that we have to find ways in which we prioritize it in the same kinds of ways that we prioritize hospitals. We don't ask questions about Sick Kids Hospital consuming too much energy. When we think about the baby that was just on the news the other day, it was pronounced dead in one hospital, and then they found out it was still alive, and they carted it to Sick Kids to do the proper procedures there. Food production has got to be, it's a fundamental right, and there's a fundamental crisis surrounding it, has to be attacked with a similar kind of fervor. A lot of discussion at the International Rice Research Institute to have an international task force on this, actually. That includes vertical agriculture. And what I mean by this more specifically is to couple it, controversial though it may be, to nuclear power plants. I could envisage a scenario personally where large tracts of food production towers would be constructed in places that are far from civilization, in the Canadian North, coupled to power plants. It could be below ground. You know, in Canadian Shield Rock, where there is safety from earthquakes and things. You don't want to do this in Japan, obviously. But yeah, energy is the final frontier, as they often say. And yet, to a physicist, when you talk to a physicist, energy is not limiting. They'll tell you energy is everywhere. The problem is we've got to keep it out, right? So the harnessing of energy responsibly, in combination with the mandate for food production, yeah, that's the ultimate problem with this approach.
Can I actually, Franco, go ahead, please.
First of all, thank you for this absolutely terrific talk. And for the record, and Malcolm can validate this, if I was doing it again, I would become a plant biologist. So
It's never too
That's what he
late,
maintains to
Frank.
me all the time. I always figure he's pandering to me.
Not
but
making that up. I think it's phenomenally interesting area, just in terms of the interconnectivity and the interdependencies that you have so wonderfully articulated. A question I had was around the developmental trajectory of the rice plant, and if you divide up the stress effects in terms of the actual mechanisms underlying the stressors at the early stage of development of the plant versus later stages, can you just comment on where is the more vulnerable period and what are the longer term consequences of early stage?
Yeah, that's an excellent question, actually. It touches upon a research direction that we're only beginning to go into with rice. We have some data that suggest vulnerabilities at the very early stage being fundamentally different. A plant that is just germinating with a tiny seedling, as opposed to even 3 weeks in, let alone when it goes all the way to yield. So I mentioned some of the data earlier that produce the larger rice plant or the one that consumes much less water. To our positive surprise, actually, the developmental biology played in the favor of our trickery in the sense that by the time grain set occurred, the advantages that were already visible in the younger plant were, however, much, much larger. You could not have anticipated allometrically, using simple models, the size of that advantage. And likewise, disadvantages cannot easily be extrapolated from studying, say, only a young rice plant. There's a common problem in all of biology that you have to focus on a developmental stage. How old will my organism be when I study it? And how applicable will what I learn about it be to the older organism? So yeah, we're quite cognizant of this, and there's quite a bit of effort now. Lasse has begun some experiments on this front as well. Wayne does a lot of this kind of thing, looking at developmental stages and what drought tolerance or salt tolerance or nitrogen benefits might mean there.
I'm going to actually draw questions to a close at this point in time and finish on 2 points. And one was one I wasn't going to make, but the last 2 questions have really driven home to me the wonderful value of this particular institution and the event that we're participating in today. When you have a leading English scholar and a leading neuroscientist asking questions and engaged in discourse with a leading biologist and having meaningful exchange, it really says something about the maturity of the institution in which we participate and the way in which we create fora to talk together. And that's one point I want to end on. And thank you all so very much for being here and participating in that particular discourse. The second point I want to finish on is to bring us right back to where I started, which was about quality and quantity. Some of you are undoubtedly familiar with Robert Persick's book, Zen and the Art of Motorcycle Maintenance, where he grapples with the issues of quantity and quality and the way in which we deal with that in academia. We tend to measure things in simple metrics. Herbert, for example, is a Canada Research Chair. He's the founding director of the Canadian Centre for Global Hunger Research. He is someone who sits on the editorial board of 3 journals. These are metrics that we're able to put a number to and say, this is what makes this individual, at least as far as we're concerned in an academic institution, a wonderful individual. But I would say that it's the quality. It's what we've heard today, the ability to take simple fundamental concepts and convert them into translatable ideas that actually have resonance the world over. It's that quality that really makes a difference. And I really can't thank Herbert enough for such a wonderful talk today, where he's showing us that quantity indeed is important, but it's really quality that makes a difference. Thank you very much, Herbert.